want to thank Dr. Delaney and Dr. Sim. It's really an honor to follow them. I think as you probably gathered from their talks, we've come a long way in the last couple decades in terms of the treatment of chordoma, in particular advances in surgery and radiation. And I think that is in no small part to the two speakers and the groups that they work with. Mayo Clinic and Mass General have really been leading the charge along with a couple other institutions to push the envelope and have made really tremendous advances in surgery and radiation that are leading to better outcomes for chordoma patients. Unfortunately, we believe that there's still more work to be done, more room for improvement. You saw some of the survival curves that Dr. Delaney and Dr. Sim showed, and frankly, all of us at the Chordoma Foundation, even though we see the improvement, we still find that to be unacceptable. And if you look at the kind of overall population of the United States, unfortunately, chordoma patients in general are not doing as well as those that you saw necessarily on the screen. The patients that get to a center of excellence like Mass General or Mayo Clinic or elsewhere tend to do better than the average. And so overall, the recurrence rate and the overall survival is still unacceptably low. And so we have a lot of work to do to find therapies that can help prolong and improve the lives of patients, regardless of where they are in their journey with the disease. So that's really a big part of what we do at the Chordoma Foundation. It's core to our mission. And I'm really excited to share that there is tremendous progress being made, especially in the last several years, towards better treatments, and we hope ultimately a cure. So I'm going to, in the next half hour or so, try to give an overview of the progress that's being made and where we're headed in terms of advancing the development of new therapies for chordoma. So first of all, I think it's important to kind of understand the problem that we're facing. We, about 2 years ago, undertook a survey to try to understand the challenges and situations that members of the chordoma patient community face. And we were really tremendously pleased with the response that we got. In fact, many of you in this room may have participated in the survey. We had over 700 responses from patients and family members, which represents a huge proportion of the chordoma patient population. What we found was somewhat mixed. We found that 40% of patients reported being disease-free, which is a wonderful thing. But the flip side of that is that 60% of patients were not disease-free. They either had residual tumor after a prior treatment, or they actually had progressive disease, or in some cases metastatic disease. So if you look at the proportion of patients who have either advanced disease, metastatic disease, or progressive local disease, it represents a little over a quarter of the patient population. And so if you think about that in the United States, we believe that there are about 2,500 chordoma patients. And so that means that there are at least 600 or 700 patients at any given time in the United States that are in need of therapies beyond surgery and radiation. And that number that continues to increase every year. We also tried to understand not only to what extent patients, their tumors were controlled, because ultimately the goal is not to treat the tumor, it's to treat the patient. It's to make us well so we can get back to living life as usual. Unfortunately, these results were really quite stark, and really a wake-up call that even if we can achieve good tumor control, patients are being left with significant quality of life issues. So you see over 40% of patients in our survey report having chronic pain. Over a third, in part because of the chronic pain and other difficulties that they face, report depression or anxiety, difficulty walking, fatigue, difficulty sitting, balance impairment, double vision, etc. Some of these effects are caused by the tumor. Others of these effects are caused by the treatments. And so even though we're making progress on developing surgical and radiation approaches that are improving survival, improving disease control, there's still a long way to go in terms of improving quality of life. And so as we think about the Foundation's research agenda and the work that we're doing to push forward new therapies, it's not just about controlling the disease, it's about improving quality of life as well. I think that's very important to keep in mind. So, at the end of the day, what do we all desire? What are we working towards? We want treatments that will improve quality of life, treatments that will reduce recurrence. So, even with the best surgical and radiation therapy approaches, unfortunately, sometimes there are recurrences. Sometimes a tumor is, it presents very large or presents late. And as you heard from Dr. Delaney, they may not be a candidate for surgery. There needs to be therapies that can stop progression of those tumors. And ultimately, we want to cure the disease, meaning that we stop the disease from progressing and such that it never comes back and never threatens the life or health of patients again. So when we started the Chordoma Foundation in 2007, I'll be honest and say that we didn't know how to do this. And really, I think no one did. There were just a handful of researchers working across the world, for the most part in isolation. There was no coordinated, systematic effort to try to improve the treatment for this disease. And so when we started the Chordoma Foundation, the very first thing that we did back in 2007 is we tried to bring together all the world's experts in chordoma, everyone who is doing research on this disease, and as well as people who had made progress in other types of cancer and other diseases, to see if we could bring their expertise to bear on the disease. If you look hard enough, you'll see Dr. Delaney in this picture. He was there with us, and I believe has been with us at every single chordoma research workshop since 2007. So I think that just really speaks to his level of dedication. But this was kind of an incredible gathering. It was the first time that any of these investigators had ever come together, and I think Dr. Delaney can attest the atmosphere in the room was really electric. There was a tremendous amount of excitement, ideas flying left and right, frankly. We, at the end of this two-day meeting, left with a very clear sense of what the important unanswered questions were, what the key research priorities were, and some sense of how we needed to take steps to address those priorities. And so it became clear over, following the conference that we needed to put together a medical advisory board and a scientific advisory board to continue to guide the Foundation's research efforts over time and to continue to refine the plans and the priorities that came out of this research conference back in 2007. And based on the outcome of that conference and with the help of our scientific advisory board, the Foundation put together a research roadmap, which essentially has guided our research efforts over the last 9 years now. And I think the important thing to note about this research roadmap is that it is comprehensive. It spans the entire treatment development continuum. Because the reality is that in order to achieve our ultimate goal of finding better treatments and a cure for this disease, there's a lot of important research that needs to happen in coordination, in tandem, across a wide variety of different disciplines. But the important thing to note is that each of those, the research in each of those domains or each of those disciplines is important, it's critical, but not by itself sufficient to solve the problem. What's required is progress across this entire continuum to ultimately lead to better treatments and a cure. And so I'll just kind of at a very high level walk through the kind of contours or the key areas of this research roadmap to describe how the Foundation has tried to go about advancing research in a systematic fashion. So at the very beginning, in order to really do any research, to understand the disease, to have any hope of developing better therapies, researchers basically need a set of tools to work with. You need tumor tissue to study, you need cells to be able to experiment on, you need ultimately animal models to be able to test your therapies. And as I mentioned in the introduction, when the Foundation started and back when we had that first research conference, those resources just didn't exist. And so if you are a researcher interested in studying chordoma, your hands were really tied. The work that you could do to understand the biology of the disease and understand potential ways to treat it was really hamstrung by that lack of resources. So one of the very first things, the very first step in this research roadmap is developing those resources to give researchers the tools that they need. So there you see in that first column, resource development is the foundation upon which everything else is built. Once you have the resources to work with, then the name of the game is trying to understand what makes the tumor tick, and important, finding vulnerabilities that could make it sensitive to some of those targeted therapies that Dr. Delaney and Dr. Sim were talking about, or unique characteristics that could make it amenable to attack by the immune system. So is there some sort of defining characteristic of chordoma? Is there something about the cells that looks or behaves different that you could train the immune system to recognize that as being a problem or being foreign or disease and go and attack the tumor? That's what target discovery is all about, understanding what are the unique defining characteristics or vulnerabilities of the disease that could create an opening or an opportunity to attack the disease. So that's what scientists are really good at doing. There's labs across the world who have developed tremendous technologies that are being applied to other types of cancer. And our job as the Chordoma Foundation and as the community of researchers that we work with are to apply all of those technologies and the brightest minds to try to understand those therapeutic targets in chordoma. Once we've done that, then that kind of takes us down one of two paths. So either you have a therapeutic target for which there is a therapy that already exists. So you heard Dr. Delaney mention Gleevec, for example. Well, the reason why Gleevec is sometimes used to treat chordoma is because the target of that drug, the protein that it interacts with, was found to be overactive in chordoma. So Dr. Delaney mentioned it. It's called PDGFR, platelet-derived growth factor receptor. That is a kind of a driver of the tumor, and that targeted therapy, Gleevec, stops or interrupts that signaling pathway. So that is one example of a drug that is approved so that's on the market already, that could be applied to treat chordoma when you understand a key driver of the disease. So there's about 150 cancer drugs that are already on the market that could be applied to chordoma if we find the appropriate target for them. There are at any given time about 1,000 cancer drugs that are in development. So there are billions and billions of dollars being spent all over the world to try to develop new cancer drugs that are at various stages of their development. And the great thing is that they represent the ability to go after or to target lots of different types of proteins or vulnerabilities in the cell that even approved drugs don't yet allow us to target. So the universe of potential therapies that already exist or in development are pretty numerous. And that gives us a real head start. That means that it's possible that the typical drug development life cycle from going from idea to drug in patient is a very long, costly, and tumultuous endeavor. It can take $2 billion in 15 years, and there's a great deal of frustration with how long and how expensive that endeavor is. But we can short circuit that because there are a lot of therapies that are already in development that we can take advantage of and kind of piggyback and apply to chordoma. That being said, we have to be guided by biology. And we don't know whether we're going to be so lucky as to find a target in chordoma that is so-called druggable, where there is an existing therapy or a therapy that's in development. And I'll talk about this more later, but in fact, the target that you saw on Dr. Delaney's slides and Dr. Sim's slide that seems to be perhaps one of the most promising for chordoma, it's a target called brachyuria, and I'll talk about this more in a second. That is not currently druggable. There aren't drugs that can currently shut down or interfere with brachyuria. And so that's where this middle stage of the research roadmap comes into play. When necessary, when the biology points us to a target for which a new drug is needed, then we can't just sit back and wait for the pharmaceutical industry to pay attention. We've got to be proactive and seed and provide funding to researchers to take the steps necessary to actually go after that target and develop new therapies. So whether it's an existing therapy or a new therapy, ultimately, to know whether it's going to be beneficial to chordoma patients, it has to be tested in clinical trials. That's as smart as we are. We like to think we are, as advanced as computer models may be or laboratory models may be. At the end of the day, to know whether a therapy is effective, it has to be tested in clinical trials. And so that's really the culmination of the research roadmap is testing therapies in clinical trials. But so I've skipped a step, which is preclinical research, and that is to say that before one embarks on a clinical trial, before one invests significant resources in a clinical trial, before one takes the risk of exposing patients to a drug, and frankly, no drugs are without risk, you want to know that the therapy has a really good chance of working, that you have very strong rationale, both from a molecular standpoint. So does it make sense scientifically that the target that this therapy is going after, is that present in chordoma? But you'd really like to know, you'd like to have some model of the disease to know whether in cells or in animal models of chordoma, whether this drug will actually stop or reverse the progression of the disease. And that's where preclinical research comes in. Preclinical research basically means testing therapies or testing drugs in cells or animal models of the disease. And that's a really, really critical step because it's needed to provide the justification for going forward with clinical trials. And what we found several years ago, as the research started to progress through that roadmap and we started to identify therapeutic targets and promising therapies, what we found was that there was kind of a bottleneck because they couldn't be tested in mice. The infrastructure and the models, frankly, just didn't exist. And so there wasn't enough scientific rationale to convince drug companies and to convince some investigators to embark on those trials. And so we had to find a way to overcome that hurdle and create that preclinical data package, that preclinical evidence to justify a clinical trial. So like I said, it is a multi-step process. Research in each of these areas is critical, not by itself sufficient, and we have to proactively make sure that research is moving forward in each of these areas, and that the outputs of research in one area feed into the research that goes on in other areas. I think this is a way in which the Chordoma Foundation's approach is somewhat different or non-conventional compared with other foundations. The kind of typical way that foundations operate is to accept proposals from researchers and to review those by a group of esteemed scientists and researchers and try to select the best ones and then provide funding to those, which is important and a very necessary thing to do. But the problem with that reactive approach is that sometimes it results in missing research that is really critical. So you might end up with a bunch of proposals that come in the target discovery bucket or the resource development bucket. But if we're not focused on proactively driving forward research across that entire continuum, at the end of the day, you'll do a lot of good research, but you're not going to find a cure. And what we want to do is find a cure. So we have to be very proactive and systematic about advancing this entire research roadmap. So we have a strategy, a three-part strategy for doing that. The first part of that is to bring together a vibrant community of chordoma researchers, scientists, doctors, drug companies to facilitate collaboration, data sharing, partnerships. We find that there is so much potential within the chordoma research community between doctors or researchers who have ideas or some resource that enables the work of someone else in the community. So just by bringing together researchers, we're able to unlock tremendous potential and facilitate projects that otherwise wouldn't be possible. Additionally, as I alluded to earlier, it's really critical that researchers have access to the fundamental materials, the cell lines, the animal models, the tumor tissue to do their work. And historically, it has been quite difficult for researchers, not just in chordoma, but across many different fields, to get access to those materials. And so the Chordoma Foundation tries to act as essentially a one-stop shop. Well, you could think of it like an Amazon.com, if you will, for these materials. It is surprisingly difficult in the absence of a centralized repository to get access to these materials, it takes oftentimes months, three months, 6 months, sometimes a year or more to transfer these materials from one institution to another if there isn't a centralized repository. And that is time that is wasted and delays the development of new therapies. And then finally, and very importantly, is to strategically fund research. So the reality is that this work all requires significant amount of investment, but it requires a smart investment. We recognize that we are a relatively small organization with a modest budget. The Chordoma community is relatively small. We have to be very, very smart about how we spend our money and deploy our resources. We recognize that we're never going to be able to invest the kind of money that's invested in lung cancer or breast cancer or some of the more common cancers. So we have to be very smart and strategic and highly leveraged in how we invest our money. So to create this community of research, a vibrant community of researchers, we've done a number of things. We've hosted 5 international research workshops now, which have grown every year. The most recent one was in July in Boston, where we had about 120 doctors and researchers from 11 different countries, over 50 institutions. So it's so exciting to see the field growing. We started off with about 50 researchers at our first conference. So we've increased by 140% in the span of 9 years. In addition to that, it's not just enough to bring researchers together on a kind of an every other year basis. We have to be in constant communication, and that's a key thing that the foundation tries to do. We keep our finger on the pulse of what's happening in labs across the world, so that when an advance is made in one lab, we know the other labs that might be in a position to follow up on that, or similarly, if someone's running up into a problem, we can stay aware of others who might be able to help them with a solution to their problem. So we're very actively trying to act as pollinators, if you will, among different researchers in the community. We kind of always have had this sense that bringing together researchers at conferences has a beneficial impact in terms of facilitating greater communication and collaboration. But we invest significant time and resources in this, and we wanted to understand kind of from a quantifiable perspective, what was the impact, or what is the impact that these conferences have? And so to do that, we actually, before and after some of our conferences, we've surveyed all of the participants to get a sense of how they know or have interacted with other participants of the conference. And the results are really quite striking. So on the left side of the screen, you see basically a network diagram showing all of the relationships between participants at the conference prior to the conference. And then on the right side of the screen, you see all of the relationships between participants after the conference. And I think you could, just looking at it visually, eyeballing it, you can see that there's a significant increase, but you can also start to quantify that. So the number of collaborations among the participants of the conference in the year after the conference increased by 19%. The number of relationships among participants increased by 65%. The network density, which measures essentially how many of the potential relationships within the network are actually realized or actually become real, increased significantly. And then degrees of separation. This is the Kevin Bacon concept. We all heard 6 degrees of Kevin Bacon. Well, in the research community, the same can be measured. The same applies. We've decreased, just after one conference, decreased the degrees of separation between any 2 researchers in this network by 23%. And so you can imagine that this doesn't translate directly into better therapies for chordoma patients, but it's an important step along the way. You can imagine, if a researcher has more relationships and knows others in the field, when they've got an idea or they've got a problem that they're facing, they now, after coming to a conference, they have others that they can now talk with and compare notes with. And what we found is that this has really started to produce some really impressive and exciting collaborations, multi-institutional collaborations between researchers at different medical centers. To the second part of our strategy in terms of developing the supply of scientific resources that researchers need, this is an area where we've really focused a lot of our efforts over the last 9 years now, and really was the primary initial focus of the work that we did. So we recognized that until this problem was overcome, until researchers had the cell lines, the animal models, and the tissue to work with, it was going to be very difficult to make substantial progress. So we've developed centralized repositories of tumor tissue, of cell lines, and of mouse models, which are available to the research community. We set a goal back in 2007 of acquiring certain numbers of these resources. We wanted to acquire 250 tumor samples, 10 cell lines, 10 of a particular type of mouse model, and 1 of another type of mouse model. The reason why it's important to have multiples of these resources is because in reality, every person's tumor is unique. Even though there are commonalities among chordomas, every tumor is different and can behave slightly different. And if you are only studying 1 or 2 or a handful of these models, then you can easily be misled because each model is representative, for the most part, only of the tumor from which it was derived. My PI at Duke, Dr. Kelly, whose lab I worked in, was fond of saying, all models are wrong, but some are useful. And I think that that's a really important thing to keep in mind. Cells in a dish or cells in a mouse are very different than tumor cells in people, but oftentimes they're useful tools for helping to understand how the disease works. But you need large numbers so that you can really be confident that the effects that you're seeing in the cells or in the animals are actually legitimate, that they actually represent the biology of chordoma and biology that's shared by many or all chordomas as opposed to some quirk of one individual's tumor. So I'm really, really pleased to say this is tremendous. It's taken a tremendous undertaking, in particular by my colleague Patty Cogswell, our research manager, who's been with us for about the last 4 years, to develop these repositories. So we've acquired 172 tumor samples from 172 patients. We've exceeded our goal of developing 10 chordoma cell lines. We now have 12 validated chordoma cell lines, which are available to the research community. There are at least 17 more in development, and our scientific advisory board now has said, let's keep going because there are so many different subtypes of chordoma. We want to expand out this set of models so that they are even more representative of the entire spectrum of the disease, to make sure we've got recurrent tumors, primary tumors, metastatic tumors, tumors that are from pediatric patients and adult patients, pre-radiation, post-radiation, male, female, every different permutation you can imagine. We've also made substantial progress towards developing mouse models, and we have a very robust pipeline of new mouse models that are being developed. And I'm really excited that we actually just heard at the research conference in Boston in July that perhaps the first genetically engineered mouse model has been developed. And I say perhaps because it's still very early. There are mice that have tumors. They look like chordoma. They behave like chordoma. But they're still being characterized. And so we don't want to declare victory yet. But this is tremendously exciting because what it means is that we understand the biology of the disease enough now to be able to give it to a mouse. So we can actually induce chordoma in a mouse now by replicating the genetic alterations that are found in chordoma tumors in people. Those same genetic alterations, when introduced into a mouse, leads those mice to develop chordoma tumors, which is just incredible proof that the science is working, that we're understanding the key drivers of the disease and able to translate that into a model. It's really, really spectacular. Just to give you a sense, though, of the kind of persistence and perseverance that effort takes. So I left the lab at Duke in 2008, and I kind of worked there part-time a little bit through 2009. Right when I was leaving the lab, this project was getting underway in 2009. So it's been a 7-year undertaking, but finally it looks like there's been a breakthrough. So then with respect to funding research, as I mentioned, our approach is fundamentally, I think the defining feature of our approach to funding research is that we're proactive as opposed to reactive. We fund research with a plan as opposed to being reactive and just waiting for researchers to come to us with their good ideas. We try to fund the best researchers across the world, regardless of where they are. We're not affiliated with any one institution. We try to go where the best science is happening. We really try to encourage and, when possible, require that data is shared in a timely fashion. We try to work hard to leverage outside funding. So if we can get research done without having to put the Chordoma Foundation's money on the table, that is a huge win for us. And frankly, we've been able, by providing cell lines and providing animal models, and just by connecting researchers with one another, making the right introductions, we've been able to leverage many times more funding than we've been able to provide in terms of outright grants. And then I think a really important feature of the way in which we go about supporting research is that we try to pick the right tool for the job. So typically, the way that foundations operate, the funding tool that they use is grants. And grants are really important and beneficial because they enable researchers at places like Mayo and Mass General and others to pursue promising projects and promising ideas and exercise their creativity, their ingenuity, to solve problems, to make discoveries. But grants are not always the best way or the most efficient way to get research done. Sometimes it's more efficient and more cost-effective to get research done by paying for it on a fee-for-service basis, by going to a company and getting it done by a service provider. Sometimes it's more efficient to get research done by offering a prize and trying to encourage many different researchers to try their hand at solving a problem. And so we've tried to be very systematic and strategic about picking the right tool for the job. So where has this led us? In terms of developing this vibrant research community that I described, we've now hosted 5 international research workshops. We are in touch with and aware of at least 300 active Chordoma researchers across the world, which when you think about it is really phenomenal. There are 300 new cases of Chordoma every year, and there are nearly as many, if not more, researchers across the world now who are studying Chordoma. It's really tremendously exciting for me to see the level of enthusiasm, the level of investment that's being made in this disease. We've made significant progress in terms of developing the scientific resources that are so critical for advancing research. And we've now distributed mostly Chordoma cell lines, because we've just more recently started distributing the mouse models and very recently started distributing on a pilot basis some of the tumor samples. But in particular, we've distributed cell lines to over 100 researchers and companies across the world. And that has enabled research that otherwise would not have been possible and has leveraged a really tremendous amount of funding that was already existent in those labs or those companies, and have been able to direct that to Chordoma research. So in total, we've supported research in 26 labs across the world. We've invested a little over $4 million now. But we estimate that the amount of funding that we've leveraged is at least 5 times that, over $20 million. And frankly, that number is probably low and is going to be increasing as we move towards clinical trials. So this has resulted in more than 30 peer-reviewed publications that have come from work that we've funded or from labs that we provided cell lines to. And this is really what's exciting. So now we have a much more developed understanding of the biology of the disease. We're by no means there in terms of completely understanding this disease. There is still much more work to be done. But we have a much better sense of what are the key molecular drivers, what are the key genetic changes, what are some of the potential therapeutic targets. And this is just a partial list that I've got up on the screen. Some of these you saw in Dr. Delaney's slides, but now there is a growing list of targets for which there are therapies that already exist, that are either in the clinic already or that are in development, which is tremendously exciting. And we are taking steps now to try to test those therapies in both mouse models and then ultimately in clinical trials. But as I mentioned earlier, perhaps the most important target, brachyury, is not druggable yet. And just briefly, what is the evidence that points to brachyury as being such an important target? Well, thanks to work that was done in the UK, a collaboration between the University College London and the Sanger Institute that was supported by the Chordoma Foundation, we now know that 97% of Chordoma patients share a misspelling or a change in this brachyury gene that is inherited. So almost every single person who has chordoma has this alteration in the brachyury gene. Now, this oftentimes raises the question, so is chordoma genetic? Well, I think this is a really important question to address. This same change is shared by 40% of the population. Okay, so this does not by itself cause chordoma. So just because you have this change does not mean that you're going to get chordoma. The way you can think of it is if you don't have this change, you're very likely to not get chordoma. But it is not by itself the cause of chordoma. It loads the gun, but something else comes and pulls the trigger, and we've got to figure out what that is. So there are rare cases of familial chordoma. There have been about 10 families in the entire world where multiple members of the family have been reported to have chordoma. So 10 out of billions and billions of people over the last 50 years, as far as we know. Exceedingly rare. Chordoma by itself is rare, but familial chordoma is exceedingly rare. But what we do know now, thanks to work that was done at the National Cancer Institute and at Duke, we do know that at least some of those familial chordoma cases are actually caused by an extra copy of the brachyury gene. So rather than inheriting 2 copies of the gene, the individuals in those families inherit 3 copies of the gene. That is a pretty remarkable finding. It's actually the very first time that a cancer has been associated with an inherited gene duplication. And so just as a quick sidebar, this is kind of an interesting thing about studying a rare disease like chordoma is sometimes it teaches us things about cancer in general that might not have otherwise been discovered. So now we know that this is a way that cancer can form, which is a pretty neat and interesting thing. We know that the brachyury is activated in all chordomas, almost all chordomas. So it is turned on to a very, very high degree. And as Dr. Delaney and Dr. Sim mentioned, this has now been replicated by several labs. But we know that if you shut brachyury off in chordoma cells using genetic approaches, that you completely stop the growth. You induce growth arrest. So that is proof of concept that not only is brachyury important for starting and initiating this tumor, but it's also critical for the ongoing survival of the cells. So suffice it to say, as I mentioned earlier, we have a very significant interest in trying to develop therapies that will target brachyury. That is absolutely critical for ultimately, we think, finding a cure for this disease. In the meantime, though, we're not waiting for that to run its course. We're putting processes in place and programs in place to try to, as quickly as possible, identify whether any of these other therapies, any of the targets for which existing therapies exist, whether any of those are really promising and could potentially warrant being tested in clinical trials. So as over the last 4 or 5 years, as our knowledge has started to unfold and has really blossomed, and as those therapeutic targets started to be identified, we had researchers coming to us, asking, I've got this idea, I've got this target that I've identified, coming to the foundation asking for funding to test a drug in cells or a mouse model. And we provided some of those grants, but what we recognized was that there were a lot of researchers across the world who kind of had a particular target in mind and who all wanted to do the same thing. But it was really inefficient and costly and time-consuming just to test one drug or 2 drugs in one lab. It took a significant amount of money. You had to hire a postdoc or a graduate student to work on the project, and you had to grow up the mouse models, and it was a very time-consuming and inefficient process. So we said, all right, what if we, rather than trying to do this scattershot, what if we said we have one lab that's going to do all these drug screening experiments, and we'll do them with the benefit of economies of scale. So we'll have all the mouse models in one lab, and if a researcher or a company has a drug that they'd like to test, it's a one-stop shop. They go straight to this one lab, they get the drug tested, they don't have to hire a scientist and import the animal models and wait a year for the animals to grow. You get your answer very quickly and cost effectively. So this is what we've done. We've contracted with a lab in Texas that services the pharmaceutical industry. The foundation has paid for this lab to grow up all of the chordoma animal models that exist. And now when researchers come to us with an idea, or when a company comes to us with an idea, or when a scientific advisory board identifies a promising therapy, we can quickly and efficiently test that in this drug screening pipeline. And here you can see that we're able to dramatically decrease the cost of doing these experiments, and perhaps most importantly, dramatically reduce the time of doing these experiments so that we can get answers faster and move on to clinical trials faster. So this is unpublished data that has come out of this drug screening pipeline. I think the really exciting thing is here is that this is data that the foundation owns. And the foundation, at the end of the day, our biggest incentive is to find cures for this disease, is to deliver therapies to patients as quickly as possible. And so, we are able, we have a real need for speed, and getting this data out into the public domain and in the hands of researchers and physicians as quickly as possible is really core to our mission. So, we're able to share this data even before it's published. So this is just an example of one of the drugs that was tested. It was identified by our medical advisory board and scientific advisory board as being really promising. And here you can see that in one of the mouse models, it basically induces complete growth arrest. So the top line is the untreated animals. The orange line is the treated animals. So based on this, our medical and scientific advisory board have agreed that this is a really promising target to go after, a promising therapy to test in the clinic. I should say that not all of the curves look this good. Some of them, the treated animals, the tumor progresses just as fast as the control animals. So, I think that this is a way to really do a reality check to say, is the science that we've done and our understanding about the disease, is it really panning out in terms of actually being able to control the tumor in a living organism? Here's another example of a targeted therapy that was tested in the mouse models. This is an EGFR inhibitor. So you heard Dr. Delaney talk about EGFR inhibitors. So there's a whole slew of them now. The one that was tested in the clinical trial that Dr. Delaney referenced in Milan, that one actually, of all the EGFR inhibitors that were tested, that actually did the worst. But of course, that clinical trial was done before we had the benefit of this drug screening pipeline. And frankly, this is kind of a quick sidebar into how the clinical trials get done. The only reason they used that drug is because that was the only drug that they could get from a pharmaceutical company. There was only one pharmaceutical company with an EGFR inhibitor that they could convince to donate the drug for the study. But by having evidence like this, we're now able to make a much stronger case to companies to provide their drug for the clinical trials. And I'm very happy to say now that there is a clinical trial that's starting likely at the end of this year, early next year, with one of these EGFR inhibitors that shows even more activity than lapatinib, which was tested in Italy. So this is really the most exciting part of all the work that we've done at the Chordoma Foundation, and really is where it's starting to feel like the progress is becoming real in terms of actually having an impact on the lives of patients. So, at the end of the day, all of the work in the lab is in service of bringing therapies to patients, and so we at the foundation have a real focus on trying to facilitate well-justified clinical trials for which there's strong scientific rationale. And I'm really, really happy to say now that we have a pipeline of 6 clinical trials that the Foundation's Medical Advisory Board and Scientific Advisory Board have identified as being high priorities and worthy of the Foundation's support. So we're supporting these trials in 3 ways. By helping the investigators and the companies initiate the trial. In the case of a company-initiated trial, it's helping them find and connect with the right sites to do the trials. In the case of the investigator-initiated trials, it's helping them connect with the company and making the case to the company to donate drug for the study. We are really fortunate to have a very active and dedicated medical advisory board that reviews the protocols for all of these studies to make sure that they are well-designed and appropriate for chordoma patients. That's a really critical step that I think it's impossible to overstate the importance of that. Not a lot of companies have done clinical trials in chordoma, and so being able to rely on the medical advisory board for guidance in terms of how to design the study is a tremendous benefit. Another really critical role that the foundation is playing in these studies is helping to get the word out to chordoma patients, to the patient community, that these trials are available, to help patients understand which trials they may be eligible for. You can imagine with a rare disease, it's very difficult to find patients for the trial, and many clinical trials, especially for rare cancers, never even finish because they can't find enough patients for the trial. And so that, what we found was a real barrier that was preventing these trials from getting off the ground in the first place, because companies and investigators didn't think that they would be able to find enough patients for their trials. So this is a really important role that the foundation plays in terms of connecting patients with trials. And then finally, and importantly, providing the funding that's needed to fill the funding gap that may exist for some of these trials. Some of these trials, fortunately, are funded by companies. Others, the investigators are donating their time or have resources available through their institution. But in many, if not most cases, there is a funding gap that needs to be filled, and that's where the foundation steps in to provide that last bit of funding to cover the non-drug related costs of the study. It's a really, really exciting time at the foundation, and to see where the research has progressed I think for all of us, it's tremendously motivating. We think, and we've set a goal for ourselves of initiating 10 clinical trials by 2020. So one of those clinical trials is already ongoing. 3 more are going to start late this year or early next year, and then 2 more will start next year. So by the end of next year, there'll be at least 6 ongoing clinical trials and more to come, which is really extraordinarily exciting. So just in summary and in closing here, this is a kind of a brief snapshot of what our current research priorities are. In the interest of time, I won't go through them in great depth, other than to say that I think over time, if you were to plot our priorities over time, it has very much shifted towards the far end of that continuum, towards clinical research. So the money that's being invested now is much closer to actually having an impact on the lives of chordoma patients. It's still very important to continue investing in developing more and better models, in continuing to refine our understanding of the disease and the biology and identifying therapeutic targets, in developing therapies that will target brachyury and running the drug screening pipeline. But now the focus has really kind of entered the realm of pushing forward this promising pipeline of clinical trials. And this work is really made possible by overwhelmingly by the generosity and investment of the chordoma patient community. So with that, I will close and open it up maybe for 1 or 2 questions. I think we're running a little bit short on time. Dr. Delaney.
From a practical point of view, I'm becoming somewhat concerned at the cost of clinical trials. And I think there are probably opportunities to reduce some of the costs. One is the adoption of a central investigational review board. Currently, many studies have to be independently reviewed by investigational review boards who sort of pile on questions. The other is we usually have contracting issues between different institutions, and one might be able to ask the NCI for some sort of standard clinical trial contract or support that, because at least from much of the agreements between institutions, and the other is advantage of electronic technology and data capture, because when you've looked as you fund institutions, it's staggering how much it costs to do some of this work.
Yeah, Dr. Lane, I'm glad you brought that up. So I think you're absolutely right, and you know certainly better than I am, better than anyone, that there are, especially when you're doing a multi-institutional trial, there is, I think, a lot of kind of infrastructure costs, transaction costs, startup costs that make the trial more expensive, but not only more expensive, but more time-consuming. And really, at the end of the day, what matters most to us is getting these trials done quickly so we can get answers quickly and get therapies to patients more quickly. So, you know, one thing that we've been talking about at the research conferences and obviously the medical advisory board, and I think there's wide agreement now, is that what this field would really benefit from is a kind of multi-institutional clinical research consortium. And so that is a hope and a vision of ours. It is a kind of a significant undertaking that by itself requires a substantial investment. And this is, I think, a real opportunity for perhaps an NIH grant. And I think we'd very much be supportive of and have tried to try to seed that effort. Unfortunately, NIH funding is not what it once was, and there isn't a great deal of funding available for these sorts of efforts. I think the funding call is only available every 3 years or so. But so there's potential role there for NIH grant, but I think there's also a role for philanthropy to try to seed that effort and kind of get it to the point where a group of investigators, a group of sites could come together and put forward a really compelling proposal to the NIH to get this sort of consortium funded in the long term. So that's definitely something that is on our radar and something we'd very much like to do. And obviously would welcome the opportunity to work with physicians at Mayo and really anyone who might be interested in pursuing that.
I just wanted to make a comment, excuse me, that all of us Chordoma survivors would be in an entirely different place without all the efforts that you've put forth, Josh, in getting the foundation going and being brilliant in developing the collaborative work that it's taken to get there. So thank you.
Well, I appreciate that.
Yeah, sure.